The recent discovery of an ancient molar, a tooth that most likely belonged to a young archaic girl who lived up to 164,000 years ago in a cave in Southeast Asia, provides new evidence that the mysterious human lineage known as the Denisovans, previously only known from caves in Siberia and China, also lived in Southeast Asia, according to a new study. However, it could also be evidence of a Denisovan-Neanderthal hybrid who lived before the last full interglacial, 130,000 to 160,000 years ago. When it came to Neanderthal expansion into East Asia, it may have been that aggressive territorial expansion coupled with an aggressive mating strategy was an effective evolutionary strategy to temporarily fend off their modern human rivals, the Homo sapiens. The Denisovans' exact location is debated. The fossils discovered so far have all come from mainland Asia. Now the newly discovered tooth could be the first fossil evidence of Denisovans in Southeast Asia. Any additional fossils identified as Denisovans are important for better understanding their biology and evolution. Scientists discovered the tooth in 2018 at Cobra Cave in Laos's Annamite Mountains, which has an entrance about 110 feet, 34 meters, above ground. The limestone cave, officially known as Tam Ingu Hau Tu, was discovered due to its proximity to another site where previous research had unearthed ancient fossils of modern humans. Animal fossils found in Cobra Cave included rhinoceros, tapirs and sambar deer. The tooth was a molar that had yet to emerge from the left side of the lower jaw. This suggested that it belonged to a child aged 3.5 to 8.5 years. Analyzing the dirt and rock surrounding the tooth using techniques such as luminescence dating, which determines how long mineral grains have been exposed to sunlight to estimate their age, and radioactive dating, which determines the age of things based on how long it takes certain chemical elements to radioactively decay, revealed that the molar was between 131,000 and 164,000 years old. The study also confirmed that the tooth belonged to the Homo genus by analyzing proteins in its enamel, and the lack of proteins associated with the Y chromosome suggests that the tooth came from a female. The researchers did not examine the fossil for ancient DNA because this genetic material rarely survives well in the type of sediment found in the cave and under the tropical conditions found in Laos. In fact, when scientists compared this molar to teeth from other hominins, the group that includes humans, our ancestors, and our closest evolutionary relatives such as Australopithecus, they discovered that its internal and external 3D structure resembled Neanderthals, but fell slightly outside their known range of variability. Furthermore, the tooth was distinct from those of modern humans and Homo erectus, the first known human species to use relatively sophisticated stone tools. Although the scientists were unable to rule out the possibility that it belonged to a Neanderthal, they concluded that its close physical similarity to a Denisovan specimen from China indicated that the molar was most likely Denisovan. The tooth indicates that Denisovans lived in Southeast Asia, which is important for understanding their distribution. We know their DNA got there because it is found in recent Southeast Asian groups, but this also suggests that the population lived in the area. This also demonstrates that Denisovans lived in a variety of environments and latitudes, adapting to harsh conditions ranging from the cold mountains of Altai in Russia and Tibet to the tropical forests of Southeast Asia. Even if this new fossil is not Denisovan, any new human fossil from a region where few ancient human fossils have been discovered, such as Laos, is significant, especially if it is a non-Sapiens fossil, as this appears to be. The new findings may shed light on how different human lineages coexisted. Neanderthals lived in Europe and Western Asia, while Denisovans inhabited much of Eastern Asia alongside other humans. However, it is unclear whether, when and where all of these extinct groups may have met. These findings indicate that other fossils in Asia should be reanalyzed using modern techniques. Genetic studies suggested that Denisovans were adapted to high altitude and cold climates, but we now know they also lived in warmer, more humid climates at low altitude. Even though recent genetic studies have suggested that Denisovans and modern humans met in southern Asia during the late Pleistocene 40,000 years ago, researchers did not expect to discover a Denisovan tooth in Laos, but according to the paper, the differences from Neanderthals that they observed do not preclude the tooth from belonging to this taxon, and would make it the southeasternmost Neanderthal fossil ever discovered. The alternative hypothesis that the tooth belongs to a group of Neanderthals that made an incursion into Southeast Asia cannot be outright rejected, even if it is less likely, according to the paper. What's more, some paleoanthropologists have suggested that the marble skull from China represents an Eastern Asian Neanderthal, primarily because of its purported age, rounded orbits, and the presence of robust supraorbital tori. If the reconstruction of the marble skull is accurate, the upper face morphology of the marble one is similar to Neanderthals, with a prominent nose and thick parietal bone. Although we don't have a precise measurement of cranial capacity, 1,300 cubic centimeters is the estimated cranial capacity assigned to marble 1. This is in a range similar to modern humans and Neanderthals' cranial capacities, and the fossil is possibly as young as 130,000 years. In another study, researchers suggested the apparent diversity of supposedly unique human forms during the Middle Pleistocene is the result of a complex network of cross-continental interbreeding based on the 140 to 120,000-year-old Nesher Ramla fossil from the Levant, which features a mix of Neanderthal and primitive Homo erectus traits. Therefore, the evidence for Neanderthal hybridization is extensive and cannot be ruled out. Indeed, the analysis of the nuclear DNA of Denisovans points to a sister group relationship with the Neanderthals. Thus, it is possible that the archaic humans in Asia were a mixture of Neanderthals, Denisovans, and an already widespread Asian erectus population. In fact, geneticists found evidence that 3% of the Neanderthal genome came from archaic humans, and estimate that the interbreeding occurred between 200,000 and 300,000 years ago. Furthermore, 1% of the Denisovan genome likely came from an unknown and more distant relative, possibly Homo erectus, and about 15% of these archaic genes may have been passed down to modern humans who are alive today. Thus, these findings confirm previously reported cases of gene flow between ancient humans and their relatives, and also point to new instances of interbreeding. This is a complicated history, but what we are learning is that the interactions between different human species in the past were much more convoluted than we had previously appreciated. Indeed, it has long been believed that Neanderthals originated and flourished on the European continent. Yet recent morphological and genetic studies have suggested that they may have received a genetic contribution from a yet unknown non-European human group. This contrasts with the traditional view that considered the European continent as the sole place of inhabitation of the Neanderthals and their direct ancestors. For example, ancient bones from Siberia, known as Denisova-11, are the only known physical remains of a first-generation human hybrid, the offspring of a Neanderthal and a Denisovan. As remarkable as her remains are, a thorough examination of the genome of modern-day Homo sapiens reveals that there were many more hominin hybrids around. The finger bones were discovered in the Denisova cave, a cave in Siberia's Altai Mountains that has yielded numerous remains of Denisovans, Neanderthals, and Homo sapiens. The remains, which are estimated to be around 90,000 years old, were given the name Denisova-11. According to the journal Nature, scientists performed a DNA analysis on the bone and discovered it belonged to a teenage female with a Neanderthal mother and a Denisovan father. Nevertheless, this only scratches the surface of these interspecies relationships. Further genomic analysis revealed that her Denisovan father had a small amount of Neanderthal ancestry from hundreds of generations before his birth. Neanderthals and Denisovans are two species of hominin that are closely related to us. They both lived in Eurasia until they were displaced by modern humans approximately 40,000 years ago. Neanderthal remains have been discovered throughout Europe, the Near East and Central Asia, but Denisovans have only been found in the Denisova cave and a specimen from the Baishia karst cave on the Tibetan plateau. Given these scant remains, a few teeth, a lower jawbone, a couple of skull fragments and a single finger bone, We know very little about Denisovans. We don't even know what they looked like, but they probably resembled Neanderthals. Most people consider Neanderthals, Denisovans and Homo sapiens to be distinct species, though some consider them subspecies. But it is undeniable that they were all closely related enough to have mated and had offspring. Most people alive today, particularly those of European descent, still carry Neanderthal genes in their genome. It is also known that Denisovan genes persist in the genetics of some Southeast Asians, allowing them to adapt to high-altitude environments. While evidence of this intermingling is readily available in our DNA, this hybrid is a vivid and very human reminder that human evolution is a story of hybridization. In fact, recent genomic research has revealed that hybridization between divergent lineages is the norm, not the exception, in human evolution. In a related study, researchers created reconstructions of Denisovans using methylation patterns, which are chemical changes in their ancient DNA. In many ways, they were similar to Neanderthals, but in others, they were distinct. Scientists reached their conclusions after years of intensive research into DNA methylation maps. DNA methylation are chemical modifications that affect a gene's activity but not its underlying DNA sequence. The findings reveal a human figure with a low forehead, protruding jaw, and nearly non-existent chin, an overall anatomy similar to that of another extinct human group, the Neanderthals, who lived on Earth around the same time period. The scientists first compared DNA methylation patterns between the three human groups to identify differentially methylated regions of the genome. Next, they looked for evidence about what those differences might mean for anatomical features, drawing on what is known about human disorders in which those same genes fail to function. Studying Denisovan anatomy can help us understand human adaptation, evolutionary constraints, development, gene-environment interactions, and pigmentation patterns. At a broader level, this work represents a step toward being able to infer an individual's anatomy from their DNA. This allows researchers to predict which skeletal parts will be affected by differential gene regulation and in what direction, such as a longer or shorter femur. They then used this method on the Denisovan and were able to create the first reconstructed anatomical profile of the mysterious Denisovan. The study identified 56 anatomical differences between Denisovans and modern humans and Neanderthals, 34 of which were in the skull. For example, the Denisovan skull was likely larger than that of modern humans and Neanderthals. They probably shared Neanderthal characteristics like an elongated face, a wide pelvis, and also had a larger dental arch and lateral cranial expansion. Finally, another study suggests that the mysterious extinct human lineage, known as the Denisovans, interbred with modern humans in at least 2 waves. The discovery suggests that Denisovans and modern humans shared a more diverse evolutionary history than previously thought. But one mystery is why modern humans in East Asia have only tiny amounts of Denisovan DNA, while possessing Neanderthal DNA in amounts the same as Europeans. The answer could be the Denisovans interbred with Neanderthals before modern humans arrived in the region. Although modern humans are now the only human lineage still alive, others not only coexisted with modern humans, but also interbred with them, leaving DNA in the modern human genome. Such lineages included not only the Neanderthals, modern humans' closest extinct relatives, but also the mysterious Denisovans. Previous research discovered that, while Denisovans had a common ancestor with Neanderthals, they were nearly as genetically distinct from them as Neanderthals were from modern humans. The research estimated that the ancestors of modern humans split about 700,000 years ago from the lineage that produced Neanderthals and Denisovans, and that the ancestors of Neanderthals and Denisovans diverged about 400,000 years ago. Nonetheless, genetic analysis of fossils from these extinct lineages revealed that they were close enough to interbreed with modern humans. Denisovans are still largely unknown. So far, researchers have discovered only 5 fossils that are certain to be associated with them: 3 upper molars, a finger bone, and a jawbone, severely limiting what researchers know about them overall. Scientists who discovered a skull in China named Dragon Man claimed it belonged to a new species, Homo longi, but many other researchers believe it is a Denisovan skull, but it also has many Neanderthal-like features. Did Neanderthals expand into Eastern Asia prior to the last interglacial period? With that tantalizing statement, we leave you to ponder the mysteries of our shared human history. Until next time, stay curious and stay questioning. Also, please subscribe, share and explore our channel's other highly compelling videos. Thank you.